Hello, this is Mr. White. We are in the 7th grade science textbook. This is Chapter 7, Lesson 1, The Genetic Code. My Planet Diary. DNA Debut. In 1951, English scientist Rosalind Franklin discovered that DNA could exist in a dry form and a wet form. Franklin made an image of the wet form of DNA by exposing it to X-rays. The X-rays bounced off the atoms in the DNA to make the image. The image, see the background on the next journal page, was so clear that it helped scientists understand the structure of DNA for the first time. Her discovery was important for figuring out how genetic information is passed from parent to offspring. Franklin's contribution to science was not only in her research, but also in that she succeeded at a time when many people thought women shouldn't be scientists. What forms the genetic code? It took almost 100 years after the discovery of DNA for scientists to figure out that it looks like a twisted ladder. When James Watson and Francis Crick published the structure of DNA in 1953, they added another clue to how traits are passed from parent to offspring. DNA contains the genetic information for cells to make proteins. Proteins determine a variety of traits, from hair color to an organism's ability to digest food. The structure of DNA. Parents pass traits to offspring through chromosomes. Chromosomes are made of DNA, and proteins are located in a cell's nucleus. Look at Figure 1. The twisted ladder structure of DNA is also known as a double helix. The sides of the double helix are made up of sugar molecules, called deoxyribose, alternating with phosphate molecules. The name DNA, or deoxyribonucleic acid, comes from this structure. The rungs of DNA are made of nitrogen bases. Nitrogen bases are molecules that contain nitrogen and other elements. DNA has 4 kinds of nitrogen bases: adenine, thymine, guanine, and cytosine. The capital letters A, T, G, and C are used to represent the bases. Chromosomes, genes, and DNA. In Figure 2, you can see the relationship among chromosomes, genes, and DNA. A gene is a section of DNA molecule that contains the information to code for one specific protein. A gene is made up of a series of bases in a row. The bases in a gene are arranged in a specific order. For example, A, T, G, A, C, G, T, A, C. A single gene on a chromosome may contain anywhere from several hundred to 1,000,000 or more of these bases. Each gene is located at a specific place on a chromosome. Because there are so many possible combinations of bases and genes, each individual organism has a unique set of DNA. DNA can be found in all the cells of your body, except for red blood cells. DNA can be found in blood samples, however, because white blood cells do contain DNA. Order of the bases. A gene contains the code that determines the structure of a protein. The order of the nitrogen bases along a gene form a genetic code that specifies what type of protein will be produced. Remember that proteins are long-chain molecules made of individual amino acids. In the genetic code, a group of 3 DNA base codes for one specific amino acid. For example, the 3-base sequence CGT always codes for the amino acid alanine. The order of the 3 base code units determines the order in which amino acids are put together to form a protein. How does DNA copy itself? 2 new cells, or daughter cells, result when a cell divides. To ensure that each daughter cell has the genetic information it needs to carry out its activities, DNA copies itself. DNA replication is the process in which an identical copy of DNA strand is formed for a new cell. Replication is very important, since daughter cells need a complete set of DNA to survive. DNA replication begins when the 2 sides of a DNA molecule unwind and separate, like a zipper unzipping, between the nitrogen bases. Next, nitrogen bases in the nucleus pair up with the bases on each half of the DNA. Because of the way the nitrogen bases pair up, The order of the bases in each new DNA strand exactly matches the order in the original DNA strand. This pattern is key to understanding how DNA replication occurs. Adenine always pairs with thymine, while guanine always pairs with cytosine. At the end of the replication, two identical DNA molecules are formed.